Let's give it a start. Good morning, everybody. Thank you for joining this session. This is now more about diagnostics, as you know, multiparameter flow cytometry, and I would like to show you what applications this can have. So I have a mixed bag of information. I see some faces whom I know very well, and they know some things maybe even better than I do. But I try to have it mixed so that it's also for beginners and people who want to orientate themselves. So for those who are experts, please forgive me that you will hear many things you already know. Conflict of interest. So flow cytometry is a technology which dates back into the 80s with the nowadays machines which can be used in diagnostic workup, which have up to 12 to 13 colors of fluorescence. What means that you can assess cells with different antigens on the cells up to 12 parameters, or 13 parameters, which allows a very detailed information about cells and their antigenic repertoire. You all know how bone marrow, for example, and this is the favorite liquid material which we are investigating, how this is sampled, usually in children from the hips. And you also see that how the staining is done. It's the incubation of the blood cells with the antibodies, and it can be a mixture of antibodies set on the cells at the same time. And then after this incubation, the sample runs through the flow cytometer, and there is a chamber where there is one cell after the other walking through the light channel, and there is this fluorescence light sent to the cells, and then they start to gloom, depending on the other antibodies, and the antibodies are labeled with the fluorochromes, and the different fluorochromes give back different emission spectrum. And that is recorded by a computer. And then you can have these nice plots, which I will show today in more detail. You know, nowadays, I remember the time when I started, we were with a screwdriver, we were working on the machine to change things. And so this is not anymore happening. So these things works almost alone. You can put your samples in, and there is a carousel, and it works through automatically. So times really have changed, and the machines in former times were big like that, and nowadays they are tiny like that, and there are also machines you even can carry on. So this technology is moving, and some people thought this will be a dying technology, and you do everything with genetics, etc. But I would like to show you why this is not the case, and why these machines also and this technology will be there to stay. I would like to start with immunophenotyping in leukemia. Why do we do that? Is this outdated? You also hear about genetics a lot in these sessions, and why do you actually need still this type of assessment? Flow cytometry is a quick methodology. So on the same day you have the result. This actually offers many things in inadequate time, where genetic and molecular methodology takes much longer. To make a decision. So the first decision you have to take is actually to if you have a new leukemia, to assign lineage. Lineage can be assigned also by microscopy, that's for sure, but not in each and every case. In some cases you won't be able to say so. If it's an M0 or it's an lymphatic leukemia, you cannot decide by microscopy. So the lineage assignment is the active question on the first day, and it's the question of treatment choice. We will go into further details why this is also sometimes tricky and why flow cytometry really adds on it. The second thing is you can do by flow cytometry is actually the antigenic profiling. Because you could say, okay, after having the lineage, we are done, not more of interest. I will show you why this is not the case. Antigenic profiling has much to do also with what was in old times the initial step to do. It's actually to say, okay, what is the maturational classification of the leukemia? You will say there are some criteria, like AGIL criteria, for example, to think about maturation. So you could say, what the hell is maturation? Why is this interesting? And I will show you that this actually has to do enlarged with this antigenic profiling with informing about the underlying genetic lesions. And this actually can also be used for the molecular and the genetic people thereafter to really find their way better than they would, because genotyping, etc., is now a very large field in leukemia. It's not about tele-AML1 only or hyperdiploid, or also if you go for the AML, for example. It's very diverse, and you have subsets of cases, like 2% of all, you wouldn't even think this is the direction of the genome. Flow cytometry can inform on this, and I also would like to show you. So you actually can help working together with the geneticists to do a better and quicker job. Next thing is, what we are doing nowadays is minimal residual disease assessment. You all know, you are familiar. It's working in ALL, it's more and more working in AML, it's what we really have to do. So at diagnosis, you profile your sample and you will know what markers, what leukemia-associated markers you will be using throughout the follow-up. So this is also what you are informing at that time. Minimal residual disease clearly is about treatment stratification, but not only. It's also about informing what to do in the further workup, in the further schedule of the treatment, about targeted immunotherapies. And this is a mainstay which you will be needing for the future, which you cannot do by genetics. This is the mainstay of why you will needing flow cytometry. We have been preparing guidelines on immunophenotyping. And just as an example, for sure there will be other guidelines as well, but the IOPFM consensus guidelines have been done by a larger consortium. And nowadays, in immunophenotyping, we do it at least with 6 colors at diagnosis. We have built some recommendations what to include. Importantly nowadays, because it's multicolor, you have a backbone of markers where you really can define what are the blasts, apart from the other background cells. And then you can individually, based on the blast gate, you can investigate the blast marker profile. What is nowadays a standard is that within the sample there are residual lymphocytes, and those can be used to say where's the negative region, where's the positive region. So you have everything within the sample. This is also advent of the multicolor approach. And I will walk you through actually what the more complex things you might encounter. Blast cell heterogeneities, the thinking nowadays, what to do with when there is an MPAL, the mixed phenotype leukemias, how to think and how to work with those, because this is something which makes the so-called easygoing thing at diagnosis more tricky. So that's the current consensus panel. We have this as a single platform panel. We are running these several surface markers. All in all, you will end up with something like 35 markers, for example, in a regular panel. So it will depend on the laboratory you're working in. But this is something like a recommendation. It includes markers which really help you, myeloid markers, lymphoid markers, help you to find out, okay, what kind of lineage it is. This actually is defined also by WHO and HL scores. But then, and also maturational level, some of those markers are of interest also for immunotherapies, like you see here, CD33, which you always will also, although it's a myeloid marker, you will always also investigate on lymphatic leukemias. But then you have recommended other markers which go more into the direction of markers for immunotherapies, for MRD, LIPE, leukemia-associated immunophenotype assessment, or, for example, to define the genetic backgrounds, like markers like CRLF1, CRLF2, and CD371. I will touch that. That's, for example, a panel we are using in our daily workup. So we have combinations of myeloid markers, T lymphoid markers, B lymphoid markers against the background of panleukocyte markers. So we easily can track different cells and compare the expressions and build the negative zone, the positive zone. So it's just an example. It's not how everybody should do that. What is also nowadays of interest, in former times, marker expression was rated in percentage. Like the marker is positive in 22%. You mean 22% are very strong and the rest is completely negative? Or you mean you have a curve, Gaussian curve, where you cut somewhere and say, okay, everything to the right is positive and the rest is negative? So what does it mean? Nowadays it's more captured in something where we say, okay, it's negative expression. It's a weak expression, but we also capture whether it's partially expressed. That's very important because it tells you it's not homogeneous. And we really refrain from giving simple percentages, but because anyway it's meaningless. It's more that you say, although this is semi-quantitative and is also some kind subjective, it helps you better understand what's going on. It's weak, it's strong, or it's partial. Let me come to the dominant lineage assignment or lineage assignment. In former times, it was thought that B, T, and myeloid leukemia is very well separated. Now we learn that there are myeloid markers on conventional ALLs and the other way around. So the big question always was, what is actually the leading differentiation? Nowadays, also in terms of how to treat. In terms of treatment, because ALL is much more frequent, ALL markers are much more frequent on the surface, like CD19, for example. The tendency goes that a cell or a leukemia which is mostly positive with CD19 should be treated in the direction of ALL to start with ALL treatment. This is the common narrative. So it means actually that AML criteria go in the second line, meaning that actually if you have myeloid markers and you don't meet ALL criteria, you will be saying, okay, that's classically an AML situation. It's a tricky discussion on that, so don't take me plain so that it's always in each and every situation. But this is the current narrative that at least for treatment, the thinking is that you rather start with ALL treatment and then see what's going on as compared to say, okay, if you find, let's say, 3% or 4% MPO, myeloperoxidase, on something which is otherwise an ALL, should be treated as an AML. Because the MPO was in old time was something like the hero of everything. The lineage assignment is something you can do by eyes, but you also can do actually eyes. You can have this profiling, and you see nice profiles of many antigens. You see 48 antigens selected. At the end, we did in this profile 37 antigens. These were, for example, from my institution, 700 cases, and you see various B-ILL subtypes, which also can be subdivided again, as I told you. You see the AMLs, they behave with other antigens, they are positive, and the Ts also. It easily falls into parts, and when you do that, for example, with some IT tools, you also see that you easily can separate ALL, AML, and TILL. But there are some overlapping cases, for example, and about those we need to talk. As said, MPAL has been defined actually by WHO in the time before. Things developed. There was FAB morphology. Then came AGIL in the 90s. The European group of immunophenotyping in leukemia defined something which was called BAL, B-phenotypic acute leukemia. And WHO nowadays, they stepped over to take this and to build some more, let's say, condensed criteria. Meaning that, for example, if you have an MPAL, the mixed phenotype leukemia, if you have myeloperoxidase on it, as said, and markers of a T or B lineage. So with this concept, okay, how to treat? It's an MPAL. So this is why we said, okay, actually we would like to have it to inform better in what direction to treat, as I told you, and this is this dominant lineage concept. So if you go for the mixed phenotype leukemia, the dominant lineage concept sees two things. One is a simple co-expression MPAL, meaning there is, let's say, a single plus clone which has additionally markers of the other lineage. Or what you can have is bilineal or multilineal complex MPAL. And this really makes a difference. And what you have to do by immunophenotyping, find that difference. So simple is this. You have something which is B-cell differentiated CD79A or be it CD19 on all the cells, and only a subfraction has also myeloperoxidase. This is simple. Then you have bilineal, so really two different blast subsets. And when you investigate for genotype, they will have the same genotype. They will have, let's say, the lymphatic genotype. They will have the MLL, for example. And then you may have the complex. What we are meaning is complex. It goes in several directions. Only B, only myeloid, or intermediate, or even behind those green are also some red ones. The reds are the blasts, some undifferentiated. So you can have everything. So decide with us what to do. So what we did in the dominant lineage concept is actually that we review everything. We review morphology, where does it fit to? We review the genotype, we review the immunophenotype, and then come to a conclusion. I mean, it's also so that immunophenotyping is not the only thing on the world you can do. So for this, for example, this was dominant lineage. This was, although MPAL, going into different differentiation in B lineage, CD79, for example, and lysozyme, so also into the myeloid lineage, all the cells were CD19 positive. This was a simple hepatiploid ILL at the end. Was treated like an ILL, no change, nothing happened, cured as an ILL. If you look at the cells morphology, you won't say it's an ILL. Some cells might be an ILL, not all of them. They really have the myeloid features. So it's really something to put together information from all these diagnostic possibilities. We have learned that MPO by antibody, and there are different antibodies on the market, these antibodies give different signal. So one lab uses, for example, the BD antibody. You see these are ILL cases, and this is the antibody which have mostly used in the literature, which is from Under Group, for example. You see MPO is on this side, is clearly positive, and BD is clearly negative. So you will have labs informing you, okay, it's a negative situation, a positive situation. There has been no standard worldwide, no standard. The knowledge is the antibodies which you are using, you should know what they are doing. And there should be standards. So if you have a case like that, you would say, okay, this is no MPAL. If I find that, I would say it's an MPAL. So this is also why MPO alone as a single antigen is not a good information whether this is an ILL or is not an ILL. It's something like let's say, a system behind. In the most recent WHO, they reacted. Former times you have seen, 2008, they said MPO positivity. What does it mean? 21%? 3%? No, nowadays they say intensity in part exceeds 50% of mature neutrophil level, so it must be high. It must be strong. It's not anymore on low levels. So that's good, actually. Whatever, I cannot comment whether this is a good denominator, but the idea is take care. Don't take everything for serious what you find. But Also, this really exists. Bilineal, so multiple or double differentiation from the same genetic background. It goes into myeloid lineage as well as, for example, B lymphatic lineage. So that's really a problem. How to treat that? It's not just by numbers. You say, okay, well, the one are 40%, the others are 60%. This is why we say 60% is more than 40%. It's about resistance, okay? What we treat is the question about what better, what will survive. So this is, for example, a real case, and you see those more myeloid cells, while the lymphatic blasts are smaller and more condensed, and you also see that by morphology. Nevertheless, there is no dominant lineage. We are not building dominant lineage based on numbers of blasts. And this is why we should not take this case as an ILL, for example, even if it has 80% ILL blasts. Because it might happen that the myeloid compartment stays while you treat as an ILL. This is, for example, the same case. Red is the lymphatic blasts, blue the myeloid blasts. And they started on ILL treatment. So ILL treatment, day 15, all the lymphatic blasts gone. Myeloid blasts remain. Will you cure that patient with ALL therapy? Not so sure. So this is what we would call on-treatment selection. You would say, okay, let's say randomization, blinatumomab, ba. Not for randomization, please. There is well-known situation, and the friends actually who published this paper, lineage switch. They are in the room. Hello. Hello, friends from Argentina. What they found is actually there is MPAL, and we really have to take care not to overlook even tiny clones of MPAL in certain situations. MPAL exists. And these cells, they can outgrow, and they can come to an early switch. It's a switch of leukemic phenotype. And under therapy, they can come up because they are undertreated by ILL treatment. This usually goes along with MLL, KMT2A rearrangements. That's usually the case where it appears. But we also know, and this is also a paper, and the first author is also in the room. Hello. That you can have those switches as in the relapse. So not early, but later on. And in MLL situation, they can switch from ILL to AML to ILL to AML. This also happens. And this also occurs after CAR cell therapy, we have heard. So the next thing is actually, what can we also inform? So we talked about MPAL and the situation where immunophenotyping is very helpful. Then there is, for example, something where we say, okay, maturational stage relation, and also something with MPAL. This is, for example, the early T-cell precursor leukemia. It was found several years ago, 15 years ago, to be a very high-risk lymphoblastic leukemia. To some sense, this is a mixture of T and myeloid leukemia. And that is actually, we also have seen situations where actually there was even more myeloid differentiation. But the interesting thing is they really need strong therapy. And nowadays it's more or less solved, everybody says, from the ILL trial committees, but those cases usually need high-risk therapy. But what is very special with them, they don't have, in all cases, genetic markers for PCR. So also flow is important to follow those. And they have special phenotype missing some of the regular T-cell markers, and in addition, on the other hand, expressing myeloid antigens, as I told you. So we come to leukemia maturational subclassification. This is from old times. B1, so this is AGIL subclassification. B1, B2, B3, B4. B2 is the so-called common ILL, expressing CD10. And in old times, it was thought, oh, this is just maturational hierarchy. Pro-B is the younger, the earlier, and B4 will be the more mature. If you look in regular maturational stages, and here you see the B cell maturation process is actually that CD10 is on highest level in the earliest ones, not the other way around, not like in Pro-B. Nowadays, it's known that actually Pro-B is so Pro-B because it's aberration. It loses, it down-modulates marker expression. It's not like a reduplication of maturational stage. It correlates with MLL or KMT2A rearrangement. Pro-B in most cases, not in all, is actually a genotypic lesion directing finding you may have. So this is why I present you this, because we are talking about maturational stage classification. In real world, we are building information on genotypic lesions behind. We also found, for example, that pre-Bs exist, which usually in the hierarchy would also be CD10, for example, because most pre-Bs are CD10 positive, but which are CD10 negative. What do they contain? KMT2A. It's a down modulation, what happens. So this is actually, KMT2A really does a lot with the cells and it builds aberrations. It even can occur in what you would say B4. In former times, it was say, okay, if you have a B4, it's Burkitt leukemia. Treat as a Burkitt leukemia. This is not the case. There exists non-Burkitt B4. Again, especially, so they may have surface immunoglobulins on the surface. Again, there is, from the people from Argentina, I already cited them once today, they also found that there are MLL rearrangements in such cells. So it's, again, has something to do with it's maturing, but it's doing something different from regular maturation. So what we are doing is not just boring maturational stage assessment, but we are really building information on what could this be, what type of leukemia, what should we look for. We had a recent case with that, and I said, okay, please look for KMT2A. They were looking for a split fish, KMT2A, came back, were negative. So I said, don't believe you, go for MLL AF10, 1011. This is usually not found with split fish because it's a very broad range of breakpoints, and it was 1011 positive. So really, immunophenotype is there to insist on what the geneticist should do. So even more on that, immunophenoprofiling really can inform on many situations of underlying genotypes. You can see this is a cluster map done by immunoprofiling only B-ILL, and you see really some subsets really fall out. And I will show you what actually is the situation. You can build information on the various antigens, which genetic subtype goes along with which marker profile. So once I see a marker profile like, for example, I'm not going to speak now about DAX4 and CLF2, it's too easy. Single marker profile to inform on the genotype. I would rather say, okay, let's talk about zinc finger, for example, profile, which is low in CD10, also going into this Pro-B area, or having a strong CD33 signal, for example. Or you go for, where is it? Yeah, 119, for example. Also very typical situation. When it has CD10, it's a normal CD10 positive leukemia, but for example, CD99 very low, always pre-B, so B3 phenotype. So it's a very classical situation. Or hepatiploid. You will find hepatiploid by expression of CD123, CD86. So you have markers really to inform what to do. So not all countries, not all laboratories, nations have everything there. You know, you do RNA-seq and this and that, and so not all of us have that. So flow cytometry is really, on the day you get the sample, you can inform, please look for that, look for that. So if you want to do that, you can do that. Also in AML this works. We can build subgroups. It correlates with FAB, with morphology. It correlates also with genotype. More and more we can go into that direction of using immunophenotyping. As said, there are few markers which really are pathognomonic as a single marker for the underlying genotype. For example, this is the CRLF2 fusion subtype of ALL. Single marker expression tells you that this is the situation. And also the more recently described DAX4 group of, and they are not few. DAX4 is about 8% of all B-ALLs. It's not few, you know. And you easily, the day you start, you will find that. And as also by RNA-seq, you can find, okay, this is the subgroup. But RNA-seq, it takes some while until you have it. Flow cytometry, CD371, one marker. If it's strongly expressed, you know it is there. This is a very interesting subtype. It goes along with something which is also called monocytic switch. And it's not the only subtype. There are, and this is from our friends from Prague, there are nowadays 3 typical ILL subtypes which tend to have this so-called early monocytic switch, which does not mean that you have to change therapy, because it's a switch not to a, let's say, malignant AML phenotype, but it's a switch to a more mature monocytic end-stage cell. So if you treat those and continue to treat those cases, even in situation of switch as an ILL, it doesn't matter. But please, you have to know what you're doing. So again, immunophenotyping at diagnosis helps you in understanding, okay, this is going to be a switch case. And if you find then on day 15 or on day 8 in blood many activated monocytes, you will understand that's actually the switch situation. These are blasts. If you do by PCR, you will find the sample is highly positive, although you don't find lymphoblasts. The DAX4 switch is rather protracted. It comes at day 8, 15, and then rapidly goes away. And you sometimes see mixtures of lymphoblasts and switching blasts. So they have this kind of increasing site scatter. So it means the cells becoming bigger and more vacuolar-sized. So that's actually these cells with the vacuoles, these monocytic cells. So they grow out of the lymphoblasts, which are down here, and then they tend to lose CD19. They really develop into something completely like a monocyte at the end. You won't understand that this is a lymphoblast or has been a lymphoblast. And it grows out into that direction to stay there when the monocytes are. You can do that in the dish, in the culture dish. If you put lymphoblasts in the culture dish for a week and insert some steroids, cortisone, they will end up like this. And it correlates with the individual patient. This is, for example, another one. This is PAX5 mutation. This is giving huge, rapid switch. It's a bit different from the DAX4. PAX5 is a more rare situation. But within day 15, you have 24% monocytes like this. Day 0, lymphoblastic situation. Day 15, really monocytic situation. Day 33, staying many. If you do PCR, this will be a high-risk patient. This patient has been treated high risk, has not been transplanted. Actually, if you say 7% would mean, okay, transplant. not transplanted. The parents refused. No need of transplant. Stayed cured. We have another patient like this at the moment. No lymphoblasts. Lymphoblastic MRD negative from day 15. High immunoglobulin rearrangement, but continuous remission. ILL treatment, no transplant. Very fancy subtype, PAX5 mutated. So let's go. I mean, we already are within the situation of discussion of MRD, because when we talk about day 15 findings, day 33, this MRD. Okay, the basics of MRD are starting with, you have to know something about what is the normal regeneration, what is the normal pattern of regenerating hematopoiesis, of lymphopoiesis, B-cell lymphopoiesis in the bone marrow. You got to compare. You got to compare the leukemic cells versus the background of normal cells. So this is, for example, to find the 3 stages from immature to mature cells in B-cell hierarchy. And you can have this profiling. And then comes the situation of ILL. You have these 3 stages, and the ILL, for example, is CD10 strong, but CD45 very negative. And you find here in the MRD situation, you find few cells of blasts, for example, very separate from their peers, which are the normal B-cell regeneration. Sometimes this is the normal cells, as here. This is the leukemia, and this is the MRD situation. So you could imagine this is something like, because this is a logarithmic scale, they have 10-fold number of certain antigens. So actually they have this more or less the same antigens, but in different amounts. So CD10 overexpression or CD45 underexpression. So I really like that picture, because for some things you see there is something which you would not think that it fits there, and some things are missing. So this is what leukemia is. To some extent, it's compared to normal cells, but in others it's different. There are several markers where leukemia is different from normal. So these are the normal stages, and you can build the expression levels of several markers, for example, and compare with leukemia, and you find there will be underexpression or overexpression in several of those markers. Nowadays, I mean, there is a plenitude of markers you could use, and you really got to think about, okay, do I need them all? What is necessary and what is redundant, because the redundancy can be also a theme. What you definitely can see that some of the markers which are used nowadays for MRD assessment, they correlate with the genetic subtype. So again, you can use them at diagnosis to inform about genetic subtype, like for example CD86 or CD72. But you also can use them in MRD as the markers, especially correlating with certain genetic subtypes. So what we do is we really trigger the MRD light markers based on the initial findings and, let's say, on the genetic background. So what we think for the moment is actually we have a backbone of markers which are always there. And then, as said, we select 2 per tube, the light markers according to the initial diagnostic profile. If it was a hepatoploid situation, you might select CD123 and CD86, for example. If it's serial F leukemia or DAX4 situation, you select others. So what we then do is actually, that's only how it's done. You start, you see what all are your nucleated cells in the sample, you eradicate something which is a doublet, you go for the intact cells which are nicely representing non-apoptotic cells, and then you can really go for the lineage, and the first hub usually in B-ILL is CD19, and in T-ILL is CD7, and in AML I will briefly touch that a bit later. And then you look for all the other marker combinations by visual inspection. This is how it's usually done. Nowadays there are tendencies to do this in automatic, IT-driven interpretation, but this is not the theme of today. Also, this procedure needs some kind of standards, how to work on this, and we have provided those standards over the years. And what I have to say is we have been in a growing group of laboratories working together, and nowadays it's about 56 laboratories in this IBFM flow network. And I'm really proud to have all those colleagues, educated colleagues, with us in the group. And then at the end, if you have a good selection of markers which are really discriminative, green are the background B cells, red are the few residual leukemic cells, you really can find out how sensitive this assay can be. It depends on cell numbers you acquire. The less sensitive you will be, the more, and nowadays it's thought to have 2 million cells, 4 million cells, so it depends on then what sensitivity is needed. It's not about sensitivity. It's not a play. It's not just an investigation, but this is clinical grade assay. So for clinical grade, usually we go for something about 1 million of cells, which is in usual situations sufficient to inform what is necessary to give a stratification. So as you can have seen, this is the 10 to the minus 5 level which we are touching. And then we already have started to find out what levels of residual disease are prognostically relevant. This was already in the BFM 95 study. This is only from the Austrian patients. We found, okay, at day 33, those who have higher levels than 0.1, and I showed you the sample before was sensitivity 0.003. Okay, so 0.1, we get that. And even 0.01 is something we regularly get that. And for example, at week 12, for example, everything which has higher numbers like that. And that actually didn't change with all the therapies we have been giving. These ranges of interest didn't really change. And my late friend, Pepe Basso, from Padua, they collected together with us a huge amount of samples, and nowadays this is the basis of all the IOBFM trials nowadays with the day 15. Early in therapy, you already can separate 3 risk groups. Early in therapy, it's an, let's say, more or less easy test. You have the result within a day. And also in the 2009 trial, this was established. Flow cytometry 0.1, or if more than 10%, go to the high-risk therapy. So it's still there. And for the international situation, I can show you where is it nowadays used, for example. And we know it's not only, I'm talking about IOBFM, but we know also with the altogether and in the CUG studies, it's the same situation. They are using flow cytometry to inform for stratification. So for now, for the IOBFM situation, we are using day 15. If there is no PCR available, we are using day 33 and 78 results. And there is this sister trial, which is the ALIC BFM trial, which is run in Eastern Europe and South America, which is also stratifying. They don't have PCR assay, and they're stratifying based on flow cytometry methodology, as I've shown to you. So what have we discussed until now? So flow cytometry is essentially in leukemia for this MRD situation, treatment stratification. It can be used for tailoring, meaning, okay, you find that there is residual disease also at relapse or in a special situation, you can investigate on the quality of response. But we should touch also the targeted therapy issue. And there is something to know for you that is also why it's important to follow leukemia, not only because you want to quantify the residual disease, but you want also to quantify the antigenic profile. Does it change? And yes, it changes. Leukemia is not a fixed profile. So if you do it at diagnosis, it informs you a lot, but it's not a fixed situation. for example, follow this case. This was a relapsed case, CD20 into that direction, only few cells, the rest is actually negative. You would say this is a CD20 negative case. You would say, okay, all CD20 intervention doesn't work. Okay, if you look what remains throughout the therapy is actually a residual disease which is fully CD20 positive. So it really makes sense to follow the leukemia not only for quantities of blasts, but also for profile. So for example, those blasts would be amenable to rituximab, for example, interventions. So targeted therapy is the theme. And there is something to take with us, is actually this kind of shifting modulation of phenotype. It's not only the direction I have shown to you, but during the steroid phase, prednisone really induces changes in 2,000 genes during application. So there is a shift of, let's say, diminishing CD10 expression, increasing CD45, 34 expression, increasing CD20. So the cells somehow do things which actually you wouldn't have thought they will be doing. We have to adapt in our MRD assay on that, but also they do it on a more or less regular basis, so we can really know what's going to happen. For CD20, this is a good thing because it's really frequent that CD20 is upgraded in follow-up and upregulated in follow-up, and also you can do it in vitro. If you add prednisone, many B alleles will increase CD20 expression. Oops. And as said, this can be, and we have done this also in vitro, you can have a diagnosis in follow-up and they get fully positive, and you can have then the rituximab treatment, and it's functional. So this is why, for example, the ALIC group, because rituximab is a rather cheap drug nowadays, it's well established, it's something, and this is going to be tested in the ALIC BFM 2020 protocol. It's going to start this year. They are testing, for example, rituximab early intervention in addition to induction therapy. As said, with all knowing that you are knowing now, okay, leukemias can switch, like in transdifferentiation, they can modulate their antigens based on steroid intervention. You have to learn that also with the CARs, there is a possibility that CAR cell therapy selects out those cells which can live without the antigens the CARs are tackling. So for example, this is a situation before CAR cell therapy. This is MRD, and you see those red cells, the MRD cells, were CD19 nicely positive and CD10 overexpressing, and so could nicely be followed. But then after CARs, when this came as a relapse, the CD19 was fully gone. It remains with CD10, it remains with CD22, so the other markers you see here, but CD19 was completely gone. Means that on the one hand, you have to be taking care in flow cytometry MRD assessment. If you only gate for CD19, you might miss the upcoming CD19 negative blast. This is the one information. The other information is, okay, what to do with such a case. Okay, you can assess even without CD19 that the blasts are reverting, but you also can inform on other possibilities. For example, CD22 intervention. And it's also important before you start the CAR cell therapy that you think, how good is my expression? How strong is my expression? How entire, or partially expressed? So this is why I'm saying partial expression, strong expression, weak expression is not for fun, you know? You build something on it. Means that, for example, you have before CAR, this was a case with not all cells CD19 positive. You see that's really, they were, this part, for example, here is CD19 negative, this part was positive, and then they were giving CARs, and what happened, actually, those which were CD19 negative, they were remaining, so the CAR didn't work. So it's on us to inform the clinicians to say, okay, will this be the right target? Is this the right situation to go for? So with all what I have said, we come to something which is really helpful. I've spoken about modulation, changes, everything. But there's, on the other hand, also something which is very good as compared to PCR, genetic, molecular stuff in investigating situations. Flow really can, because the phenotypes, overexpression of CD10, for example, some of those features, they are really generic. Many cases have this phenotype. So you really can use this information of how is aberrant B-ILL blasts, how do they look? You can take to investigate samples where you think, okay, it might be a leukemia. This is what we call detection of minimal disseminated disease. Actually, you don't have the diagnosis of leukemia, but you try to make the diagnosis of leukemia even in the situation where it's not full-blown. And this can help in the workup of latent leukemia, which is smoldering, so not fully established, or even in the diagnostic workup of lymphoma. And this is, so diagnosis unknown, and this is where I would like to draw your attention to. Just a few cases. This was a patient who had already 2 inconclusive biopsies of a destructive sinusitis, coming to us with something with appearing like a tumor. You could imagine this might be a rhabdomyosarcoma or whatever. Is not so frequently it's a lymphoblastic situation. But in any way, we found in the blood something where it's a tiny population of CD10, very positive, bit elevated side scatter, so as a sign of vacuolization. And we suspected it will be B-cell blast, and we directed the biopsy for that, and it was. Next situation. We had a case of pancytopenia with myelofibrosis. You would say, okay, it's coming from the MDS situation. So, and Ayami is there. Hello, Ayami. You would usually say myelofibrosis, thanks. We can register. Histology, really severe myelofibrosis, also in biopsy. It was pancytopenic. And what we found, actually, we found B-cell blasts in the periphery. Then we did the bone marrow. It was B-cell blasts in the marrow. Typical, so CD45 underexpression, CD10 overexpression. We went then for sorting, and we developed the genotype, and it was classical leukemia, and was treated as leukemia. Within 2 weeks, no fibrosis, regeneration of counts. It was the right direction. Flow cytometry informing about the right biologic background. Next thing, hypereosinophilia. There is a situation in ALL, which is a rare subtype, the 514 leukemia, which comes along with hypereosinophilia. It may be 10, 50,000 cells, 80% eosinophils, and you really say, my God, I mean, they're getting cardiac problems, etc. Can I start with steroids? What should I do? You have to look by flow, you have to look for blasts, for B lymphatic blasts, because if it's the 514, you better give them chemotherapy. So this was, for example, the case in that. It was 5.6. If you look here, it's a very mixed marrow, so you don't see it's really an ILL marrow. But by flow cytometry, you can pick them. Other case, pancytopenia of unclear origin, was thought to be maybe infection, maybe CMV or something. And we found, in March, we found B lymphoblastic leukemia cells in the blood. At that time, they said, it's okay, we make it control. We don't believe you. In January, and see the time lapse, in January, it was there then with a full-blown situation. So actually, if nowadays they would believe me. But the thing is, nowadays we would be able to sort, etc., and to prove it. So you are really, it's not about one day, it's about months you can work better. And then you think the situation of a child, and we come to lymphoma, where I say, okay, you get in a lymphoma patient, huge thymic tumor, pleural effusion, big neck with really shortness of breath and can't lie down, and you say, okay, my God, how am I going to have the diagnosis? Can I do the intervention, narcosis? What to do with the narcosis? How do I do it? Do it with flow. You look into, because you know T lymphoblastic lymphoma is CD99 very strongly expressing, and it's a very typical situation, and you have in blood, you have almost 1%, and you send your people, you're done. You're done. This is the diagnosis. It's very clear. And you can sort it, and nowadays the lymphoma people, they want to do some fancy genetic tests to put them on the correct risk group, and you sort them. You sort them out from the periphery. And you sort them, put them on the genetic test, and they get the sample for genotyping. So this is, I mean, I'm not saying do it with each and every patient, don't puncture them, don't make a biopsy, I'm not saying this, you know. But in special situation where there is this threat of clinical situation, Friday afternoon, it's better to do it like this. And the patient cleared within a week, as we all know. Steroids are fantastic. If you say then, okay, we do the histology on Monday, you will be too late. But we did it by flow. So it also works in AML. For example, this was a case of a Down syndrome baby, 7 months of age. It came along with thrombocytopenia. There was nothing known from the previous months. So it's not like you know it's a TAM, but it's unknown. It came with petechia and then was tested, thrombocytopenia, what is it? Okay. Can it be CMV, for example? The first clinical thinking is in a baby, it will be CMV. We said there are CD34 positive cells in the periphery. This may happen with babies. There's nothing bad about that, but they are CD11A negative. This is M7 or TAM. If you are earlier than 6 months, it's TAM. If you're older than 6 months, it's M7. They said, nah. What did you find? 0.5, we can make it a... So we all know, in former times they have been called MDS, you know, it's MDS-like feature because nothing happens for a long time. You wait, you see, they are growing, so the cardiac situation improving if they had some... So in April, in September, then you see how that was coming, you know. So we know it already from months before. It's going to be an M7. We are going to treat that patient at the end. So briefly, and I have only a few minutes, about AML MRD, because this is also now more and more coming into the focus. We have developed dual tubes with very standardized profile. Those dual tubes are to assess leukemic associated phenotypes, but also the stem cell compartment, we call it, or CD34 compartment. And nowadays it's necessary also in international collaborative group because MRD is more and more becoming a surrogate test for outcome endpoints. We have developed together with our friends in Padua an array of markers which are of interest, especially for pediatric situation. And this is what I want to show you, why these pediatric LIAP markers, which are in addition to what is known from ELN, from European Leukemia Net, which is working for the adults, why are the pediatric markers of interest? It's, for example, the values, because they are really good. CD99, 48, CD371 are really valid markers to follow in their combination on the leukemic situation. So that is a diagnosis in follow-up. You have good combinations, simply speaking. And if we standardize, and it's nowadays possible by standardizing the machinery and the cocktail tubes, we really can run this MRD testing in several centers at similar quality. So we distributed samples with MRD content to, for example, to the friends in Padua and doing at our site, and you really see it's nowadays possible really to have to come along to a very standardized level of investigation. How do we do that? We also have standardized gating strategies, and we investigate CD34 positive and negative cells, because in myeloid differentiation, it really can go into more differentiated 34 negative compartments. But what I would like to draw your attention to is the CD34 positive compartment. It's not homogeneous. CD34 positive cells are really a mixed bag of cells differentiating in different directions. And it's very interesting to look at this mixed bag, because it also follows some kind of directions. If you look with me at that chart, let me see, at that chart, starting from here, there are the most immature cells, and they can go into two directions, from here to there. These are the most mature, the GMPs, the granulocytic monocytic precursors, and so they develop into those directions. Here there is more the lymphatic area. And as said, the early erythroid megakaryocytic precursors coming from here and also from here, and the MPPs, the multipotent precursors, which have in one cell they can grow into granulocytes, they can grow into monocytes, they can grow into erythrocytes, they can grow into dendritic cells. So this is really this MPP fraction, and we really separate those by this marker combination. And we really like to look also at regenerative situations in AML MRD, because I can skip that, because it's really helpful to understand the leukemic background. The classical assumption was, and which is actually not true anymore, that there is early a separation in the myeloid progenitors and in lymphoid progenitors. Forget that, please. That's not true. The newer assumption is that you start with a hematopoietic stem cell, you come to this MPP, this multipotent cell, and then you go into two directions, which is rather the erythroid, megakaryocytic, and granulocytic direction. This is the direction defined by positivity in CD371. And the other way around is going into the lymphatic and dendritic cell direction and monocytic direction, LMPP. And there it separates again to go into really lymphatic or back also, and this is very interesting, it goes by both directions, it goes into granulocytic, monocytic precursors. So this is actually what we represent with our plot, which we have defined by these two double markers. You really can have this way or that way. And we really, by that, we can quantify the repertoire of differentiating CD34 positive cells in a sample. And if we compare, for example, leukemia, different leukemic cases, they are really doing, every leukemia does what it does, you know. It's some look like GMPs, for example, some look like LMPPs. So really, some fall out of the regular gates, for example, because they underexpress CD38, for example, or overexpress CD99. So this can really be used. This chart can really be used as something like, let's say, a different from normal aspect. So we are not looking at leukemia-associated phenotype in a way there is a single marker helpful, but we are looking at where in this chart do they appear. Why is this also interesting, for example, in Ayami's EVOC thinking? For example, we had something which was a hypocellular RCC and diagnosed in 2017, and it was a very stable situation, and we were thinking that the counts were not too bad, and so there was just wait and see situation. But But what we then did is actually we went into with the flow cytometry and saw, okay, there is a complete skewing of this regular, normal pattern, you know, with those 4-chamber view. I call it 4-chamber view. But these 34 positive cells, they were only in one chamber. Is very unusual because usually, as said, they come from here, they go from here. So if there is nothing, how can there be something? So this is actually a sign of aberration, a sign of which has to do with the disease. And so what we did, we sorted them out and we actually found that it was a chromosomal aberration in those cells. So we can inform by flow cytometry what's the underlying situation. Even if you don't find, we didn't find those this del7q because it's too few cells, you know. So you may overlook them in the normal chromosomal assessment. Another case, it was a pancytopenia, chronic severe pancytopenia was what maybe is an ITP, chronic ITP. But also had a macrocytic anemia, so it's less so an ITP, so it goes also again into this thinking of MDS situation. Maybe we tested for several inherited bone marrow failure situations together with the friends in Freiburg, and we had significantly reduced cellularity, very few megakaryocytes. So what we looked at is actually in bone marrow and blood, we looked at the appearance of the 4-chamber view. And as said, this is the normal 4-chamber view in marrow. This is the normal 4-chamber view in blood. You always have CD34 positive cells in blood. They usually don't have GMPs, but they have frequently early EMPs and MPPs and CMPs. This should usually be there. In that case, there was a complete skewing. These were missing. The lymphatic, they were there. So it's not that all CD34 positive cells were eradicated. This is strange when you think there is an immunologic process ongoing, because usually it kills everything, all the cells. But there is a skewed, some cells are still there in the marrow as well. They develop also to GMPs, but here is much less. So by this skewing, we started to think maybe this is a selective process. Maybe there is some cytokine situation missing, because as said, cytokines really are the drivers. If you say erythroid, megakaryocytic, so what happens? And what we did then, we did an extended whole exome sequencing in our institute, and what was found is it was an MPL missense mutation in the thrombopoietin gene, and so it was actually the diagnosis of a CAMT, congenital amegakaryocytic thrombocytopenia. So always when we see a skewing of some cells residual in the CD34 positive compartment, some are missing, we always start to think there is something ongoing which is not a simple immunologic process, but could be also an underlying genetic cytokine-driven situation. Other case, and I'm coming to the end, also, RCC-like situation, patient with 46 XY chromosomes, many karyotypes investigated, unfit for SCT, was a secondary MDS, wait and see. Then we looked into our chart. To some extent, it was mimicking something like normal with lacking in the LMPP chamber, but then outside, aberrant phenotype, outside the regular appearance of cells, so CD38 negative, sorted those cells, found a fusion. Last case, very strange. I remember this. He was coming along as an MPAL myeloid B, thought to be FIB M1. Had a monosomy 7. At that time, we were thinking, okay, should we transplant? What should we do? At that time, it was not known what all genotypes may be behind M7. It's rather old picture. At that time, decided to give it an interfund hybrid protocol with some modifications. reached the CR after induction, but with somehow delayed regeneration. And going through all these therapies, it was tolerated, but always regeneration was delayed. At the end, the girl finished maintenance therapy. Nowadays, is 20 years old, is happy, is in good shape, is in continuous CR. But in the first bone marrows, we found after having this regeneration, we found something which is weird. It's something like the half pattern of this four-chamber view. Actually missing something where we say LMPPs are missing. LMPPs are missing means you don't have lymphatic cells, you don't have dendritic cells, monocytic cells. You start thinking. And then we said, once the mainstay papers from the EVO group were coming along, that GATA2 situation exists, we were saying, bingo. This is the case of the patient. Everything fits together. It has a leukemia. It has a weird leukemia. It has a monosomy 7. It has poor B-cell regeneration. We didn't find any B-cells since then. We didn't have only low monocyte counts, and they don't have no LMPPs, no B-cell precursors, etc. So this is the GATA1, GATA2 window. If you have this four-chamber view and you don't find LMPPs, no PCP1s, I know it's a GATA2. So we already recently have tracked patients, and we really can know, okay, this is good. So as I've shown you, when there is a missing here, you got to think about, for example, CAMT. When you are missing here, you got to think about GATA2. So this four-chamber view is really instrumental to inform about underlying genetic disease. So I'm not going to run you through, but this is a really interesting paper you should read about GATA2 and all the other underlying SAMD9 and SAMD9L syndromes. With that, I would like to end. I'm thanking you that you stayed with me that long, because I overtook it much, but I think and I hope you have heard some things of interest for you. And many people with whom I'm working together, but most importantly, I would like to address my thanks to my group, my technicians in the lab, but also the group, the geneticists in our lab, in our institution, Oskar Haas, Karin Nebral, Stefan Körer, but also the people who are doing the whole exome testing about Karen Botstuk. And so with that, let's go for the coffee. Thank you. And if there are questions, I'm still there.